one beam. And so what I learned from that experience was that while the total dose of light that you throw on the cell is an important metric for the health of the cell, a far more important metric is the instantaneous intensity that you throw at the cell. So what that says is that a line is better than a point, and a plane is better than a line. And so you want to spread the energy out as much as you can. It also says the standard technique biologists use for live imaging, which is the confocal microscope, may be the absolutely stupidest possible way to do live cell imaging. Because not only do you have these cones of light going in and out that create all this damage above and below, at the focus you have this actinic spot of light which is going and leaving death and destruction in its wake as it drives along. And so you really need to spread the light out. So that got us thinking about why stop at 7? What happens if we start looking with more beams? The problem is now you got to worry about what happens when the side lobes of those beams get close enough together that they start interfering. And so we started modeling what would happen there. And what you find is that there's certain specific separations of these beams where you have a magic thing happen where all of the side lobes destructively interfere with one another. And you get rid of that problem, and all the excitation gets truly confined to the single plane. It spreads the energy out in that plane, and you have great modulation for doing Motz's structured illumination technique. So it's sort of a triple win. Those happen extremely rarely in life, okay? I can say, but it was wonderful. The reason I was kicking myself, though, is because these magic separation periods where this happens were basically, they represent two-dimensional optical lattices, which was that goddamn theory I had developed 10 years before in 2005 when I was trying to get back in. And so once I realized that these were optical lattices, I was able to dust off all of that old theory I had developed 10 years before and then apply it now to this light sheet microscope. And so this is what we call lattice light sheet microscopy. And you can use it in one of two modes. You can either use it in the structured illumination mode, which is the high resolution mode you see here, where you see the filopodia a little bit better, or you can dither it a little bit back and forth to smear it out to just get a plane. And here you have to take 5 images per plane. Here you only need to take 1 image per plane. So it turns out in the end to be about 7.5 times faster. It is so non-invasive that anybody who's done live cell fluorescence imaging will tell you there's a very limited amount of time, even in 2D, with confocal that you can look at a cell. But with lattice light sheet, if the protein is expressed heavily enough, we can look at it forever. The natural turnover of the protein is actually faster than the rate at which we lose fluorophores to photobleaching. And so it's really an amazing tool. One of the amazing things that it's great for is one of the other skeletons in the closet of the whole single molecule field is that it's really limited to very thin samples because, again, remember, you have to see the weak signature of a single molecule, and if there's a bunch of other molecules that are out of focus, they create a whole bunch of background and makes it difficult. So you have to look in thin parts of the cell. You can't look at cells in mitosis. You can't look at zebrafish and things of that sort. But the lattice light sheet is so thin that it's thinner than the depth of focus of the detection objective. So it means that only molecules that are in focus are excited. So you have fantastic signal to noise in arbitrarily thick specimens in order to do this. So this has been very useful for my colleagues at Genelia who study transcription, where they're able to look in this case, in a spheroid of mouse embryonic stem cells, and then be able to study the binding kinetics of these transcription factors to the DNA. But the other thing, although I swore I would never do localization microscopy again in 2008, is the other thing it's good for is getting around that nasty problem that I said about getting enough dye in your system to be able to get high resolution by palm. In the same year that we published the palm paper, Robin Hochstrasser at UPenn published a related idea where you don't use photoactivation to turn on the molecules. Instead, the molecules are whizzing by in solution and they're going so fast that they're just a haze. But if they end up binding and sticking to the part of the sample you're interested in, then they're fixed and then they look like a glowing spot. So that's called paint. And so the beauty about paint is that in a normal palm storm, whatever localization experiment, You put your labels in at the beginning and you're done. That's it. Whatever label density you got, that's what you got to work with. With paint, the whole media is labeled, so this infinite army of molecules can keep coming and coming and coming. The problem with paint is the whole fucking medium is glowing. So it's difficult to be able to use this method at high SNR. But the lattice light sheet is perfect for it. And so this is an example where you're looking now through a dividing cell that's about 20 microns thick, or about 10 times more than typical in localization, and 300 million molecules localized, which is about 100 times more than a normal localization image. So instead of looking like a bunch of dots, which is typical in localization, it actually looks like an EM image because there's enough information there to see what's happening at high resolution. And you can do multicolor imaging with this as well. But I'm tired of looking at dead things, and so the beauty of the lattice light sheet is to study 3D dynamics. So this is an example of looking at a dividing cell where the orange is the chromosomes, and the colors, the fireworks, represent tracks of the ends of growing microtubules during the division of these cells. And so from a technical point of view, this is impressive because this is 1,000 3D volumes between the two different colors of the same cell. And cells, when they divide, are incredibly light sensitive. They want to shut down so that they don't create replication errors if anything goes wrong. So they have all these mitotic checkpoints. But this cell went through 1,000 3D volumes, which represented about 300,000 2D image planes, and divided perfectly normally, and divided again after that. The daughters divided. And so it's really a non-invasive tool for studying very delicate processes inside of cells. And in this case, we're able to actually show that the velocities of these growing microtubules change as the cell goes through different phases of cell division. Let me start that again. The other thing, if the process isn't super fast and you have extra time, you can use that time to do multiple colors. So this is 3-color imaging of 3 different organelles inside of the cell. So one is the chromosomes, one is the mitochondria, sort of the power cells, the power packs of the cell, and the other is the endoplasmic reticulum, which is where proteins are sort of processed after they during their creation and folding. And so what we find is during cell division, this is a whole field of cells. Now we've taken that 3D field and sliced it into 2-micron thick slabs so you can see what's happening inside. And you can see that what happens is the endoplasmic reticulum, which was that net I showed you in that early SIM image, instead folds up and creates sort of these little pockets or cisternae. And inside of these guys, the mitochondria, which are normally long sausages, fragment and they fit inside of those pockets, and they're carried along by those pockets as the cell divides. And so that's the kind of information you can get. So working from single cells now to cell-cell interactions, this is now looking at a T cell, which is the kind of cell that fights infection. But in this case, it's sensing an antigen-presenting cell, which makes the T cell decide whether it's the right T cell to fight the infection that's happening. And so now I showed you an early example of that in SIM, where we were looking right at the boundary of this thing at high resolution. Here it's the fraction limited resolution, but we're able to see the entirety of the cell. We're able to see this, identify this very fast flow of actin away from the immunological synapse, and once the synapse is established in these guys. Now, one of the motivations for studying cell motility is metastasis in cancer. But normally, a lot of cell motility we study is on immortalized cells on cover slips, which is not exactly the geometry inside of a tumor. So another nice thing about the lattice light sheet is now to finally study cell motility in 3 dimensions in a real matrix. This is a collagen matrix here. This is with Dyke Mullen's group at UCSF across the bay. And so this is a neutrophilic cell, and you can study its motion through the collagen mesh. So the working up the next length scale, this is now looking at C. elegans, which is a model organism, a small nematode worm, and looking with our collaborator Josh Bembenek at a protein which is normally thought to be just a remnant that's left over, that's this thing, after cell division, sort of a cytokinetic remnant, and finding that it's actually co-localized to other parts of the cell. And Josh believes these remnants actually have a later role in development of the organism. Some things in the late stages of development of C. elegans, right before hatching, the muscles are going like crazy. And it's just too damn fast to get a 3D volume, but you can park at a single plane and then still image at that plane at fast enough speed to see what's going on. So what I would like to do for the future of my group is, frankly, to take cell biology away from the cover slip, because cells didn't evolve there. They need to be studied at these speeds and at these resolutions inside of the organisms in which they evolved. And there's a problem with that, is that as our light goes in, for example, a lattice light sheet, this is looking at an adult C. elegans, and these are the meiotic chromosomes. When the light sheet starts, it's great, and you can see these guys fine, but over here, the light sheet has been scrambled by the refractive index inhomogeneities of the specimen itself. Just like the water on your windshield scrambles the light coming in to your eyes. And so the same thing happens on the detection side, that once we create a light sheet, the nuclei that are closest to the detection objective we can see fine, but the ones whose light rays actually have to pass through the organism get scrambled. And so that's a big problem. These aberrations are a big problem. The good news is that we're not the only ones who have had to face this problem, and astronomers have been dealing with it for 50 years, the military and the ophthalmologists as well. And so the solution that they came up with is something called adaptive optics. So you have an astronomical object that's 10 million light years away. In the last 100 miles, when it hits the atmosphere, that flat wavefront is distorted into a distorted wavefront. In the AO system, it bounces off a mirror. You pick a little bit of the light off, you put it into a sensor that determines the nature of that deformation. But then you put it in a computer, and the computer changes the shape of this mirror to exactly cancel those wavefront distortions to create a flat wavefront and get diffraction limited imaging. So this is what you see of a star here without AO. This is with on. Without AO, the biggest ground-based telescopes in the world would be no better in seeing capability, much better in light collection capability, but in angular resolution, no better than a high-quality reflector you could have in your backyard. But with AO, the best ground-based telescopes in the infrared kick the ass of the Hubble Space Telescope. And the next generation scopes, like the 30-meter scope and the European Extremely Large Telescope, will be fucking amazing with the AO that they have. And so you gain not only resolution back, you don't gain resolution, you recover resolution, and you recover signal. Because instead of all the light going everywhere, it comes to a focus coherently and interfere. This is showing Neptune without and with AO. So how can we use this in biology? Well, there are two ways that we've done in my group. The first is exactly steal from astronomers. And I give a talk about once a year about the historical connections between astronomy and microscopy. And the moral of the story is microscopists are the retarded stepchildren of astronomers. And astronomy does everything about 50 years before the microscopists finally catch on and do it. So I just want to be a little bit quicker on the uptake as a thief and try to steal stuff a little faster. So what they do for AO generally is, because the light from that distant galaxy is not very bright, and so it's not got enough signal to give you a good measurement of the wavefront error, they shine a laser up into the stratosphere to excite some sodium atoms to create an artificial star by fluorescence up there. And then that one is bright enough, and because it's right next to the thing you care about, then a correction on your guide star, as it's called, will give you a good correction on the object that you wish to look at. And so in 2011, a group in ICFO in Barcelona said, well, what if we used a 2-photon laser? Because in a 2-photon laser, you don't get that cone of light exciting fluorescence everywhere. You only get fluorescence at the focus. So it's exactly analogous to that. So for transparent organisms like the zebrafish, which is a model organism I absolutely love, we can apply that. And so this is an example looking near the spinal cord region of a zebrafish embryo 3 days post-fertilization. This is showing the resolution and the signal without the adaptive optics, and that's when you turn the adaptive optics on. So now this is an example. One of the problems, though, in microscopy, and it's sort of a similar problem in astronomy, is that you can correct in a region, but depending on the application, in the mouse brain, a single correction can cover 100 microns. But in the zebrafish, it's big enough and there's enough variability of the refractive index across the organism that you have to create many corrections. But this guide star method can do that. In this case, it was 20,000 different AO corrections. We're going deep into the midbrain region, about 250 microns deep. That's with the adaptive optics. This is turning off the adaptive optics. So that's what a normal biologists think they're doing diffraction-limited imaging when they use their confocal and 2-photon microscopes in developmental biology. But man, they're really far away. And so with the AO, you can actually get back there. And so, and in this case, we were using the 2-photon mode of it to then study the development and the motion, the neurite-guided motility of these oligodendrocytes, sort of the glial cells inside of the zebrafish inside of that. But this is slow, and this is damaging, and this will bleach. So the thing that's on our plate right now and is about ready to see first light is a lattice lightsheet microscope that combines adaptive optics for both the excitation to keep the light sheet good and the detection to see the light that comes out. When we have that instrument, and that can also be combined with the SIM or with the localization, then we will have a tool that will really allow us to be able to study cell biology on its own terms. And I really think we are on the cusp of a true revolution in cell biology between the genome editing tools, such as Jennifer Dwalde here at Berkeley has developed, coupled with the non-invasiveness and the high speed of the lattice lightsheet microscopy, coupled with Motz's SIM microscope, we will be able to see cells as good, if not better, than we ever have before, but see them in their true physiological state, with native levels of expression, with minimal invasiveness from the light. And get everything we want, and see all the cell-cell interactions that really are the true reality of the way cells work. And so I think we're really on a cusp of revolution there. So that's fine, and that works for the zebrafish, but the damn problem is that Genelia, 80% of what we do, if not 90%, is systems neuroscience. And while we do finally, thank God, have several people there studying zebrafish, for many years, all we were doing was fruit flies and mice. And if you particularly look at the mouse brain, it looks like this. I liken it to trying to image through tofu. So trying to image neurons through tofu, because of all of the scattering that goes on, it would be like trying to image a star when the star is behind a cloud. Well, God damn it, the astronomers haven't figured that one out yet, so we can't steal from them on that. So we have to come up with our own solutions. So I hired a woman crazy enough to come and work on that problem, and that was Naji. So we had no idea when we got started as to how we would solve that problem. But we came up, I don't have time to tell about the solution, but we came up with some solutions that would allow us, outside of a guide star, but doing other means, to be able to figure out how to do an AO correction. So this is looking at structural imaging, about 200 microns deep inside of the living brain. So you put a window inside of the skull of the mouse, do your imaging through that window, and then look at the neurons there. And then look at also the functional imaging. So there's calcium indicators that light up when the activity of the neurons are going off. And you can see that you can see much better signal and noise, the activity of what's happening inside of these neurons. So that was the initial work that we did up through 2010. And then Na was able to waggle that into getting her own group outside of Geneva. And she's taken this technology light years beyond what we were able to do at that time. She gave a talk earlier this afternoon where she's applying it to study how the information that comes from first the retina and the algae and then goes to the visual cortex, what layers it goes to and how that's processed to get directional selectivity inside of the mouse. And a lot of that would be impossible to be able to see these weak signatures of all of these things without the adaptive optics to be able to pick them up. So the final thing that I want to say is, well, first I want to say something to the young people. Is one of the things, there are many, many things that disturb me about academia, as you may have guessed over everything I've said. But one of the things is it seems like people get too wound up in titles, and they get too wound up in prizes, and things of that sort. I feel like a hypocrite for accepting the Nobel Prize. I really do. I think most prizes are actually, I think the pain and the sting of not being on the winning side far exceeds the pleasure of it. And really, in the end, it's just fucking subjective, okay? It's a few guys in a room. It's their opinion. There are many opinions, okay? And so the thing that really matters is the accomplishments, you know, being able to do something or really seeing other people being able to use it. And so Nobel Prize or not, you know, I will consider myself when I'm on my deathbed a failure if these techniques are not able to answer biological questions. And I will never know enough biology to do that myself. So it means I have to get these tools in the hands of a biologist. And that tech transfer problem from something that works in my lab to something that can be turnkey to work for a biologist so he can focus on the biology rather than his goddamn microscope not working right, it's a big problem. That valley of death is something that everybody in technology faces. And so we're taking a multi-tiered approach to that. So at the simplest level, you know, we've worked with, in my group alone, we've worked with over 35 different labs who've come to work with us to apply our microscopes to biology. And in between 2013,